Recording in progress.
Good morning, panelists, excellencies, invited speakers, and also the audience. Welcome to the second days of the ASEAN Biodiversity sections, which is under the International Academic Conference on Multidisciplinary ASEAN Plus in the age of the disruptive societies. My name is Tachai Pusiyana Wins, Assistant Professor from Faculty of Science, Rangkampeng Universities. I will be the moderators for supporting and taking care for the morning sections. In this conference, we organize in the hybrid system, which include the on-site at the Faculty of Science, Rangkampeng Universities, and also the online via the Zoom systems, which is you can access into the link that we share to you. In this conference, we have the participants and invited speakers from the several countries in these regions. I think this is a very good opportunity for us to learn from our experts in this event. And we have discussed several cases from the ASEAN related into the marine biodiversity protections, coral reef, and also the flowers and other aspects from yesterday's. For today's sections in these mornings, we will have about 6 invited speakers to share and present the relevant and interesting topics on biodiversity. We will spend time for about 20 to 30 minutes for each topics, and you can discuss or you can ask the speakers during the sections. For the online audience, if you have any questions, you can raise your hands in the Zooms, or you can open your microphone to ask the question direct into the speakers. Or you can put the question into the chat box, and then we can respond you and on that one again after we finish the presentations. Okay, before we start the sections, I would request from all the audience, can you turn on your cameras, and then we can have a group photos before we start. We can take about maybe a few seconds. Our team will capture your photos. Can you turn on your camera for maybe a few seconds? Okay. 1, 2, 3. Okay, thank you. Okay, thank you very much. Okay. Okay, thank you. Thank you very much. Okay. And I think this is the time to start for the first topics, first invited speakers. We will start by Assistant Professors, Dr. Ekapan Kraichaks. Now he is the head of Department of the Biology from Kasetsart Universities. And Dr. Ekapan earned his PhD in the Integrative Biology from the University of California, Berkeley. And his experience and research areas in the plant ecology and molecular systematics. And today he will talk on the topic of the diversity within diversity, story from lichens and their symbionts. Okay, Dr. Ekapan, if you are ready, I would like to invite to start your presentations, please.
Thank you very much for introducing me, and thank you, Dr. Thamasak, for inviting me to this event. And as someone who study a lot of things, including lichens, really humble to be here, especially when the host is Rangkampeng University, who's hosts one of the best lichenology labs in Asia and if not the world. So thank you for having me here. And since there's so many groups of people and different organisms we talk about here. So in this talk, we will not go into very specific of lichens, but actually to introduce you to the wonderful world of lichens. And I'm sure that many of the audience here will agree with me that this is one of the very unexplored area of biodiversity. So in this 20 minutes to half an hour, here I would like to tell you what is a lichen, which sound like a very simple question, but not as simple as it sounds. And then whether or not the symbiosis or living to the fact that multiple organisms living together inside a lichen body does it make really successful, like, and why, and who else are actually in that symbiosis, in that relationship. And finally, to catch up with the theme of this meeting, what would, like, how much do we know about lichens in Southeast Asia or in ASEAN in general? So like the chairman say, if you have any questions or comments throughout the talk, feel free to put it in the chat box, or if you want to discuss directly after the talk, I'll be happy to respond to those. So let's first start with a very simple question. What is a lichen, right? And if you spend enough time in the forest, you see this kind of stuff pop up all over the place, especially in the tropics that look like plants, not really a plant, not really a mushroom, not anything, but this is called lichens. And lichens have been recognized for the past 400 years. And for the longest time, we don't really know what the lichens are. They just look like a plant, and they was moved to the algae, moved to the fungi, and at some point was put in with the slime mold, and they're all over the place. But with the invention of light microscope, we now know that lichen is actually a composite organism. It consists of the fungal partner and the algal partner. So it's not just one thing, it's at least two things living together, right? In this, and this living together thing is caused like having developed different morphology that looks like something else altogether. But for those people, for those who study lichens, actually, it's a bit more complicated than that, because we don't study them as a pack. And most of lichen systematics view lichen as a fungi. So if you think of fungi, you usually think of fungi as a saprophytic organism that digest other materials and absorb that as their own nutrition, right? But there's so many other type of fungi. So saprophytic is one of the major mode of fungi. Fungi can be parasitic. In Thailand, it's very popular now, right? The tanchao or cordyceps. These fungi grow on other insects and use them as their host while they're still alive, and among other things. And fungi can also cause disease in human and animals. In amphibians, a well-known one, and it can be useful one like mycorrhizal, which are associated with the roots of vascular plants, also the non-vascular plant as well. And then final mode of fungi is the lichenized version, which is the fungi that only form the relationship with algae. Only then they will have existence. So if without algae, they would not be able to make it into the world. So this group of fungi rely on the algae to make the living. And interestingly enough, that if you count the number of species of fungi all over the world, about 1/5, like 20% of all fungi are lichen forming. So it must be a very good strategy to use algae or to become lichens as opposed to be regular fungi relying on, like organic decays and all that. So what make lichen symbiosis successful? Why do fungi forego typical lifestyle and become lichens? So there's a lot of discussion about it, but the most recent paper, which just came out a month ago, and for those of you who is interested in lichens, this paper is really good, and it's a review paper on how much do we know about lichen symbiosis, and what people have agreed in the field, not agreed per se, but like what we know so far is that it's a it's a give and take relationship, and it's just not one benefit, it's multiple benefit. So the classic example that people been taught in high school and what we know so far is that, so this part is the algae, right? So here's a green algae here, and this is cyanobacteria. So both of these they have ability to photosynthesize and provide some carbohydrate for themselves, and also, so you see the little fire here is for their own metabolism, and they also provide this to the fungus to do their own metabolism. So this is traditional hypothesis is that these algae do the work for the fungus. The fungus allow them to be with each other so that they can take the food from the algae, and the algae take benefit from the fungus for the habitat and this extra niche that they gain from living with the fungus. But also another possibility is that fungus have its own carbon source, right? The fungus doesn't really need the carbohydrate from the algae, but the carbohydrate from the algae become these molecules like arabitol and mannitol, and they recognize this as a stabilizing molecule. And what do I mean by stabilizing is that it allows the fungal cell to maintain their viability during the phase of stress and desiccation. So without these molecules, the fungal cell would just rotting away. You can see that if you look at your mushroom in the refrigerator or any other fungi, without moisture, it perish very quickly. But lichenized fungi can live for longest time without water. I mean, you can ask any of the staff in the lichen herbarium at Rockham University that you found that they can stay in the dry package for a long time. And maybe the function of algae there is just to provide these stabilizing molecules. Or the third alternative is that, well, they provide this carbohydrate, the algae provide carbohydrate for fungus, and the fungus use them for both purpose, both for metabolism and for desiccation tolerance. So if you think of it this way, then it makes sense that they would spend a lot of their evolution trying to work together, try to make it work so that they have both protection from stress and also the source of metabolisms. So that's what people believe it to be successful. But all these are nice idea, but it's really very difficult to test in the lab until this study came out in 2014, and I think it was presented in Bangkok back then as well in the mycological meeting. And this is a demonstration that you can make fungus and the alga work together, even though they never need to get each other before. So this study used the yeast, Saccharomyces, and the free-living algae, Clamidomonas. And both of these organisms, they can live alone, independently, just fine. But in this study, the scientists manipulate the condition that it would give the yeast the source of carbohydrate, and this would release a carbon dioxide for the algae to use for their photosynthesis, and then in turn the algae would provide back the nitrogen back to the yeast. And when you put them together, right, both of the cells can grow really effectively. Here you can see here that if you provide a glucose and the source of nitrogen in the closed system, would allow them to grow together. But if you're missing one of them, you will have one partner here or one partner, or both partner but missing nitrogen here, or one partner and missing the glucose here. So if they are not in the right condition, they will not form such a symbiosis. But when you put them in the condition that would need each other, you can make independent organisms become dependent on each other. So with this, you can recreate the lichen-like symbiosis, and it's proven to be successful symbiosis. So that's why lichen exists, and because this symbiosis work. But how it works is a bit more complicated, because here's a study that I worked with Steve Levitt from the uh, BYU, so Brigham Young University, and we found that on the lower panel here, these are the lichen fungus, and the top here is the algae strain. So you can see that each of the lichen fungus associated with multiple algae. So it's not like lichen A always go with the fungi. Lichen fungus A go with the algae A, fungus B going with the algae B. It's not just like that. It's not one to one. It's multiple. So each species of fungus choose specific algae to work with, and from the statistical analysis, it seems that the fungal genus are like the main factor that drive this selection. So the fungus seek out the right algae to be with them, but not one algae working for them. So it's almost like a dating situation, right? You go out there, you like have like in mind, like I want this person to be my partner, and you have that in mind, but that person you want to be your partner doesn't necessarily interested in you. They like the algae can go with any other fungi as well. So it's a really complicated situation, but you can see that on the top panel here is the fungal part, and this is the algal partner. And there's like a preferred combination here that, for example, Tentapolis tend to go with a certain group of fungi, and certain group of fungi tend to prefer Tuberculosis. So there's some typical combination, but there's a lot of possibility of merging together. So that's make lichen like very successful. And interestingly, and this is getting more complicated as we go, is that different combination of fungus and algae can give rise to different things. And this is a really funny name theory. It's called the same song, different singer theory. So you imagine that, for example, we have fungi 1 and fungi 2 here, and working with only one algae. So fungi 1 and fungi 2, different fungi with the same algae, it probably keep the same lichens. So in this case, what we say the same song, different singer is that it's always produce the same lichens, even though it's different players. In this case, different fungus. Or the flip side can be too, is that only one type of fungus here and is associated with different types of algae, it still give rise to the same lichen. So in the end, we have the same song, we have the same lichen, but the players are different. On another scenario is that we have different fungus here, different fungus and one algae. And with this combination, you give rise to different lichens. So you produce different songs. So they have different lichen morphologies that come out from the same players. So the same singer, they sing different songs. And the same thing here, you have one fungus, two types of algae, you can produce two different algae, two different lichens here. And the crazy thing is that we used to think that one of these mode is possible. When I first started learning about lichens, I was told that the top left is the only way to go. But in reality, all of these 4 scenarios are possible. They are possible. So to make a lichen talus, to make the actual lichen that we see, like any type of symbiosis can happen. And it sounds like it's almost impossible to wrap our head around how different type of symbiosis can happen. So, but all this seem really fancy and good, but doesn't really make it successful. So to not prove that, but to show that it's maybe make it successful. So we have to look at the very diverse family of lichens. So, and these two are the Palmeliaceae and Graphidaceae, which also are really abundant in Thailand. and in ASEAN as well. So what are they? So if you look at this block here, you see two types of lichens on that. So the big one here that look like a leaf here, that's in Pameliaceae, and the smaller one that look like a scratch on the bark, those of Garfidaceae. So these two family, right? They are so successful. They have so many species, and the question is how, when, and how fast they diversify, and if something allowed them to diversify that much. So to do that, we create a phylogeny for these two family using Sanger sequencing data that we have, and create this molecular chronogram using molecular clock, so we know the timing of diversification. And we also try to detect when does it shift to higher diversity, right? And we use this program called basic analysis of macroevolutionary mixture, or BAM. So this give a hypothesis of like the red dot would be when it shifts to having a higher diversity. And we do that for multiple configuration, and together we calculate the likelihood and come up with this kind of diagram. And it shows that it's probably likely that the lichen shift the gear from the low gear to the high gear somewhere around here. So, and this use this technique called BAM. And in these two big family, we see a number of places that see the increased rate of diversification. For example, in Pameliaceae, you see at least three points where they increase their diversification, and this happened in the genera Osnea, Centopamilia, and Pomotrima. And if you walk in the forest enough, you see all of this all the time. And you compare that in the timing in evolution, you see that this family start to evolve, increase their diversity about 25 to 30 million years ago. And with Usnea coming a bit later and Centopamilia coming quite earlier. So what could allow this to happen, right? And the hypothesis go by that having to look at the current ecology of these two genera. Centopamilia is mostly in dry land and grow on rocks, and they believe that some of the pigments that they have serve as sunscreen. You can see that they live quite happily on the dry rock. So ability to withstand the dryness, just we talked about before, those carbohydrates from algae allows them to stay intact even when it's in the dry environment. And Usnea is the same way, right? Usnea is quite different. And in Thailand, we found this in higher mountain, it like this draping long lichens that can capture fog and moisture in the air. And this form of lichens will allow them to be successful because they're going three-dimensional. And in other family, in Garfidaceae, at least 2 points of increased diversity happened in the Garfidae and in the genus Ocellaria. And if you look at the timing of this, it's quite interesting because at the beginning of this family is coincide with the origin of angiosperm, the flowering plants. And this makes sense because both of this group are mostly on the bark. They are quite prolific on the bark, and it requires the bigger trees like the angiosperm tree as a habitat. So that's why it has to have the origin of the angiosperm first before we go in there. And for Ocellaria, it's quite similar, and especially Ocellaria because it's exclusively on bark. So it needs a lot of trees. It actually needs shaded trees. You need a forest to cover to see a lot of Ocellaria. So in the end, we found that Pameliaceae have its younger family is more dynamic, is more recently evolved, but in Garfidaceae is older but slower. But then the factor that allows them to be so diversified is that the global drying and diversification of host plants, the expansion of habitat allow these two groups of lichens to diversify very quickly. So, but I think given the amount of time, I'll probably just go a bit faster. Not speaking faster, but like going to the main details here. But all this showing that symbiosis that we talk about are highly successful and allow them to adjust to different global environment. But the question that we talk about in the field is that in the lichenology is that it's only 2 or 3 partners in there. Is it just one fungus and one alga working together? And the answer is not really. So if we look more into the details, we found that there's a lot more things in there. Even fungi, there's multiple group fungi living in the lichen. Is a multiple group of bacteria within that. So you could see, if you look at a scanning electron microscope, you see the bacteria living on the surface of the lichens here. The red and yellow dots here are the bacteria that associate with the lichens, and in the thallus, you also see the lichenicolous fungus, that black dot here. That's a fungus that live on the other fungus, and also a fungus that live inside the lichens, a different color of hyphae here representing different species of fungi. And the latest one is that we found the basidiomycete yeast that live on the top of the surface of all macrolichens. So I'll show you the video here. That this is from the study of Toby Spriebler, and do the 3D immunocytochemistry to show that there's a yeast that live on the surface of the lichens, and these are pretty consistent across the macrolichens that we have here. So, and now you could just flip onto the 3Ds, and you see that the green one are the yeast that associate with the lichens. You always find them there, and this is supposed to be a major player aside from those 2, algal and fungi. So if you draw them together, now lichens are not so much of a symbiosis between 2 partners, but it's a complex ecosystem. It's a microbiome of a bunch of microbes living together. So that's why I say it's diversity in diversity, because once you pick up one lichens, you actually pick up about 200 things in your hands, and there's so many diversity to be studied in that particular piece of lichens. But really, to end this talk, how much do we really know about lichens in Southeast Asia? And from my quick search, and this admittedly a very quick search, so not a whole lot. Actually, there are only 4 countries with the country checklist, and show between 200 species to about 1,300 species in Southeast Asia. And this number in Vietnam here, even the author, Uptrud and Sparius, admit themselves that it's very preliminary. So there's a lot more lichens to be discovered in Vietnam. And luckily for Thailand, and I speak highly of this lichen research unit at Rakampang University. They are a really strong group, and they work really hard to make sure that biodiversity knowledge about lichens in Thailand are available and keep pushing forward. So, like, I think they're one of the strongest lab in the world today for lichenology, and the resource for lichenology in Thailand are so, not Thailand, in Southeast Asia are very little. And this bibliography resource back from 1994 show only 700 references, and most of which are from Thailand. And in other countries like Myanmar, which also now under the political situation, I was shocked to read this, like only 10 species and 1 variety of lichens were reported so far. And in that study, they add 20 more. So there's a total of 30 species in the whole country of Myanmar, which is not possible, right? And in Malaysia, which cover a wide range of habitat, is having about 190 entries of bibliography, but not mentioning how many species they have. And in Indonesia, just one place alone in Papua New Guinea, they found 400, almost 500 species of lichens, but there's not much else being done in that such a big country. But if you search for the same thing, right, the lichen in Indonesia, there's a lot of interest in lichen, but not in diversity. We're interested in antibacteria, the anti-helminthics, antimicrobial activities of lichens. And that even underscore the importance of understanding the diversity of lichen first before we utilize them. So with that, I think I like to invite other people, especially for other countries in ASEAN, to look into lichens. If you have colleagues who work on lichens, encourage them to reach out to us, especially the lichen research unit in Makampang, to make sure that we push forward the ASEAN knowledge on diversity of lichens. And with that, I'll be happy to answer any question. And sorry for running a bit over time. Thank you.
Things which is the comprehensive details that we can learn from these presentations. And also this is one of the relevant topics that can reflect in terms of this conference as well. And we still have some time to have some discussions and the questions from the audience, from the online and the on-site. And do you have any questions or the discussions? Maybe you can start now. Maybe just in the quick questions from my side, just would like to maybe some details related into the environmental factors that you talk about the lichen symbiosis. And then what kind of the factors or the environmental factors that can help in terms of the promote in the formings? And then can you share that information for us?
Definitely. So one of the things that people believe to drive that lichen symbiosis to begin with is the fact that the algae and fungus moving into a drier environment. So the fact that they need to maintain the moisture or at least ability to withstand the long period of dryness, right? So the fungus itself, they need constant water, and the algae itself need constant water, but when together, they produce that extra carbohydrate that allows them to stay alive even without water. So I think that one of the primitive conditions that's allowed them to stay together. And these days you will see lichens mostly in still in the moist place, but they also be able to survive a long period of dryness. And I think that's one of the major factors that allow them to stay together.
Yeah, because I asked these questions because when you chose the map in the ASEAN and also thinking about the temperature as well, is there any effect on the temperatures, on the diversities or diversifies of the lichens? Is there any impact on that? Yeah.
Oh yeah, definitely. So if you spend some time doing field work, you recognize that lichens prefer like the environment with at least some sort of vapor moisture in the air, something where a bit more moist. And the recent work by Matthew Nelson showed that the lichens can only adjust to 1 degree Celsius of temperature increase in 1 million years. So basically, if you're asking the lichens to adjust to the higher temperature, you have to wait for 1 million years for them to adjust for 1 degree of Celsius. So with the phase of climate change we are having right now, the lichens are probably very in the bad situation because they adjust so slowly, and you can see that once the habitat changed, the forest being removed, the lichens gone fairly quickly too. So that's quite concerning. But like Southeast Asia has been a home for so many organisms before, and in its original condition, it should allow a lot more lichens than it should have today.
Okay. Yes. Thank you very much. I think in this point it's very interesting. And then also in this conference, we have the audience from the several countries in Asians. And then as the Dr. Ekapan introduced, maybe we can think further in terms of the collaborations in the more investigation in the futures. Okay. Thank you very much, Dr. Ekapan, for your presentations. And then, then we will move to the second invited speakers. Assistant Professors Dr. Nisit Ruang Sawang from Rachamankala University of Technology, Krungthep, Thailand. His expertise on the zoologies in freshwaters and marine biologies, and then he works in the several field investigation in Gulf of Thailand and also Mekong Basin areas. Today, he will talk in the topic of the biodiversity assessments in the lower Mekong basins, a case study of freshwater sponges in Thailand. And Dr. Nisit, the floor is yours. Thank you.
Thank you, Chairperson, for introducing me. Let me share the screen. Wait a minute, please. A little bit problem. Sorry, we have some problem.
Okay. You wish to spend some time, or can we move and then we can back to your sections again?
Okay. I think I can share now.
Okay.
Wait a minute, please.
Yes, okay.
ก็ต้องไม่ได้เดี๋ยวเปิดแล้ว
Maybe just for the waiting in the next sections, I would like to announce one thing. Our organizer teams for this conference, we have about the evaluation forms or the evaluation sheets that you can respond, and then we can know your feedback during the conference, and then we will drop the evaluations in the chat box, and then you can respond on that one as well. Okay.
Still have some problem, I think.
I think, yeah, we can come back to your presentation again. And I'm not sure, Ajahn Sakanan, are you available to present first?
สวัสดีครับมีอะไรไหมครับเห็นเรียกชื่อผมโทษที
Okay, อาจารย์ครับ can we have about the technical problems for the second invited speakers? May I invite you to present first for these sections?
ไม่แต่อันนี้คนไม่กด50หรือลดคนไม่ต้องการเลยชีวิตฉันอะไรวะเนี่ยมีการสำรวจ
Okay, if it's okay for you, maybe I can introduce for Ajahn Sakanan to start presentations. Okay. Then for the part of the next speakers, okay, I think Ajahn sorry for that. Ajahn Nisit, are you available to present, or shall we? Okay, we can continue with the Ajahn Nisit first. Sorry for Ajahn Sakanan. Ajahn
Sorry.
Ajahn Nisit, can you unmute your microphones and then we can start to listen your presentations.
Sorry, it's still problem about the sound. Did you hear me?
I can hear you now.
Okay.
Okay, thank you. Maybe.
Okay. Thank you, Chairperson, for introducing, and good morning, everyone. My talk today is about biodiversity assessment in lower Mekong basins, a case study of freshwater sponge in Thailand. In the present study, I compile a report on the diversity and distribution of the Southeast Asian freshwater sponges based on available literatures and a recent biodiversity assessment on the lower Mekong basin in Thailand. First of all, I would like to talk briefly about sponges. Sponges are animals in the phylum Porifera. They are sessile multicellular organisms lacking tissues, true organs, and the nervous system. The data from World Porifera database in May 2020 showed that 9,489 varied species known worldwide. Sponges are mainly found in marine habitat, about 97%, with a few species growing in freshwater. That's only 3%. Now, there are about 260 species belonging to 45 genera of living freshwater sponges. Sponges play a key function role as a filter feeders to filter the particle out of the water. They are one of the biotic components which can provide a suitable substrate for other organisms like microhabitat for aquatic organisms. They have the active biochemical potentially useful for mankind in the pharmaceutical industry. Some species can be used as bioindicator for monitoring the water quality. Let's move to the overview of the study design, which I will show from large scale to small scale. This slide shows a map of the Mekong River and tributaries. Mekong River is the longest river in Southeast Asia, nearly 5,000 kilometers length. It sources from Tibet Plateau in China to the Mekong Delta in Vietnam. The river flows through 6 countries: China, Myanmar, Thailand, Laos, Cambodia, and Vietnam. The Mekong River Basin includes 7 broad regions with diverse topography, drainage patterns, and geomorphology. The basin is commonly divided into the upper and the lower Mekong River basins. The upper River Mekong Basin comprises in the Tibet Plateau, the Three River area, and Lan Chang Basin in China and Myanmar. Now I'm focused on the lower Mekong basins. It's made up of northern highland, the Korat Plateau, the Chole Sap Basin, and Mekong Delta. It covers Thailand, Laos, Cambodia, and Vietnam. Southeast Asia harbor nearly 1/4 of planet's plant and animal species. This region is a part of the Indo-Burma biodiversity hotspot and harbor many endemic species. Discovering new species in the regions highlights the importance of biodiversity and conservation. This picture shows 224 new species discovered in the Great Mekong region in 2020. However, today, the river is rapidly changing as economic development, urbanization, and industrialization are transforming the basins. Development pressure are also creating a challenge for the country in the lower Mekong Basin. This includes environmental degradation and loss of biodiversity, hydropower development, and climate change. The lower Mekong Basin is functionally subdivided into four geographic regions. The basin covers northeastern Myanmar, northern Thailand, and the northern area of Thailand. The northern Thailand, sorry, and the northern area of Laos, last part of northern eastern Thailand, almost the entire country of Laos and Cambodia, and the southern tip of Vietnam. Although Southeast Asia is considered as a biodiversity hotspot, knowledge of freshwater sponge in this area is poor and scarcely reported. I think now the question comes to you: freshwater sponges. In general, study of Thai sponge are concentrated on marine species, and only few investigations focus on freshwater sponge. There are many reasons to study freshwater sponge in Thailand and lower Mekong Basin. The knowledge of freshwater sponge in lower Mekong Basin was still limited in biological, ecological, and application aspects. Because little is known and gap of knowledge about freshwater sponge in this area, we lack information about these neglected organisms, which are one of the important animals in aquatic ecosystem, as I mentioned before. For the field works, samples were collected by several methods, such as weighing and viewing appropriate substrate for seeking freshwater sponge, or by snorkeling, scuba diving, depending on the situation. Specimens were collected in situ, photographed, and preserved in the field by fixing in 70% ethanol, and air drying were performed. This slide shows the atmosphere during sampling collection. Interviewing the local people was also performed to collect data. We use different vehicles, such as in Thai we call saleng, from local people to help us to go to the sampling site. We cannot drive the car directly to the lake. Also, we use a long tail boat to survey in several places, such as in the picture is in Nong Han, Sakon Nakhon provinces. This slide shows GMOs, which are important traits for freshwater sponge identification. Normally, they have some mistake about the name when we found the local people told us about sponge. They call รังไข่ปลาตำแยน้ำ or หมามุ้ยน้ำ Sometimes they call ตะไคร่น้ำ or ปะการัง So we need more information to get, such as the environmental parameters to characterize sponge habitats. some physical-chemical parameter of water quality by portable digital instrument in the field, as is sampling site before collecting specimen. This slide shows ROV, a remotely operated underwater vehicle, was used to survey in the sampling area. This device is very useful. I show you the underwater clip from ROV. Why it's very useful? Because some place is very dangerous. It's not like in the marine, in the sea, because it's very clear, high visibility. In the river, sometimes the low visibility, sometimes it's zero. We cannot see anything. So the use of ROV for the first time in sampling site is better because it's very safe for researcher during sampling collection. In the clip, we also found the soft bottom in the Pong River. Sometimes we found the big size of freshwater spawn lay under on the soft bottom. One thing that I am afraid is about crocodile, also snake, also something mystery. Let's move to the laboratory works. Dry and slide collection of freshwater spawn specimens in several museums and institutes were also investigated for this study. Morphometrics analysis of important traits were performed. The available taxonomic key and useful publication were used to identify the specimen. Specimen identification was confirmed under the supervision of an expert of freshwater spawns, Dr. Renata Manconi from University Sassari, Italy. Morphological characteristics such as growth form, consistency, architectural of ectosomal and zoonomal skeleton trait of skeleton megascler, microscler, gemelloscler are considered for diagnosis at the genus and species level and were studied by light microscopy and scanning electron microscopy. This slide shows spawn surface and zoonosomal skeleton by SEM. Free and fixed gemmu were also investigated for description. This is very important thing to identification. Measurement on gemmu stress were performed by SEM. Representative specimens were compared to the original description and material from several historical collections. Now, we are show you the results in the framework of biodiversity assessment in the lower Mekong Basin, focus on mapping taxonomic richness and distribution of freshwater spawn. We discover this assemblage of spawn in this area. I report here on the discovery of new species and new record belonging to the genus Cowospongilla, Onchocercilla, Amburotora from northeastern Thailand. This paper was published in 2012. The first report from my dissertation. This is a paper from the Pong River with Onchocercilla asiatica were found in this area. In 2013, biodiversity in Southeast Asia, an overview of freshwater spawns was reported in Journal of Limnology. In 2017, the paper from Scala Biosphere Reserve in Northeast Thailand, we found Umrotora bocorensis. This is the new record from the Northeast Thailand. In this slide, we conclude that in Southeast Asia, for the overview, now we found 28 species belonging to 12 genera. For Lower Mekong Basin, there are 12 species belonging to 7 genera in the Lower Mekong Basin. In Thailand, now we found a new species in the Lampao dams. So we will publish this paper soon. Thailand now, I think we have at least 13 species in 8 genera. Description of new species and new record in Thailand indicate that freshwater sponges remain unexplored and are underestimated in their biodiversity. Therefore, biodiversity assessment in the Lower Mekong of these neglected fauna urgently needs to carry out to ensure that more species do not become declines or extinct before they are discovered. This is the ongoing work about the Thai freshwater spawn database, which was created by SCATPAD. Now we are revising this database. Last but not least, the citizen science was used to promote collaboration between scientists and citizens to collect data, create new knowledge for science, and monitoring environmental change. The purpose of this project are to create a tool to assist researchers in data and sample collection of freshwater spawn from the field and to create a support network for freshwater spawn research in Thailand. A user-friendly database, application, and data visualization were designed, containing the collection size, geographic coordinate, collection date and time, collector, photograph, color, size, growth form, substrate, environmental factors, and ecological information. In addition, basic information on freshwater sponges and a guide to collection and identification were provided as a user manual. This project officially launched at the 10th Conference on Taxonomy and Systematic in Thailand 5 days ago. Finally, I would like to thank all funding agencies supporting freshwater spawn research. Thanks my colleagues and local people for their field and laboratory assistance. Thank you for your kind attention. All questions are welcome.
Thank you very much, Dr. Nisit. I think this is quite very rare topics or the few people to work in the freshwater spawn in Thailand. And then this is quite important research areas in the future. And then thank you very much for sharing and update the information for us. And we still have some time to discuss. And then we found the questions in the chat box from Ajahn Kanokkorn. I think she would like to ask about the conservation assessment of the 12 species in the areas. Are there any high conservation concerns?
First of all, I would like to tell all of you that now we are at the first stage of the assessment of freshwater spawn in this area. But we know that some species is cosmopolitan. For example, Eunubius carteri is a worldwide distribution. So for conservation aspect, I think this species is is not have a problem in the situation, but we also need some information to know about the distribution map or the value of this bond in the for the social economics. So conservation concern, we also just like inform the local people to know that this is the freshwater spot of ฟองน้ำน้ำจืด because the education is important for the local people. Sometimes freshwater spawn have a serious problem for them. For example, the dry sample of spawn, and they will have effect of skin irritation when you touch a dry sample. Skin irritation occur. So I think we also need more study to answer about the conservation aspect. Thank you for your question.
Well, thank you very much for your explanations. That is very good to learn from you in this point. Then we have another questions from the on-site in this conference room from Professor Thamasak Yimin. Professor, yes.
Thank you, Mr. Chair, and thank you very much, Dr. Nisit, for your valuable information from your presentation. As you mentioned, freshwater sponge is one of the important filter feeders. So what I would like to ask you is that, are there any research on freshwater sponge that to show us that we can apply the freshwater sponge to solve the problem of microplastic contamination in the freshwater ecosystems?
Normally, for freshwater sponges, the biomonitoring aspect dealing with heavy metal contamination. For nowadays, I know your point, Dr. Thamasak, about the microplastic. So I did not see this point in freshwater spawn, but a very interesting topic to do research in the future. Thank you for your question.
Yes, okay. Thank you very much.
Thank you very much, Kapitan. And we have the last questions in the chat box from Ajahn Vipul Sit. Then he asks about how about the breeding biologies of freshwater sponges. Can Dr. Nisit briefly explain about this one, the breeding biologies? We cannot hear you. Can you check your microphones? We still cannot hear. I'm sure about the system.
Ready?
Okay.
Do you hear me?
Now we can hear now. Okay, please.
Okay. I forgot the question.
He
breeding
asked about
biology.
the breeding
Yeah.
biology, yes.
I perform the reproductive part in my dissertation during PhD student. They also interesting in the field, because you know that freshwater spawn they produce germu during the harsh condition. Some spawn will be degenerate. For example, in the Pong River, during in December and to February. At that time, the water temperature decreases. So spawn degenerate and this attack from the substrate. I also found the GMO they produce when in the harsh condition. We also need the new scientists, young scientists to do this aspect about the breathing biology of freshwater water spawn, because I just do only one species, the cobalt sponkilla Siam enseed in the Pong River. We also found the Panula Parenchyma larvae of freshwater spawn of this species, but we also found many species need to be studied in breeding biology of freshwater spawn. Thanks, Dr. Vipusit, for your important question. If you have student, I think I will appreciate, because I went to the Burapha in Thonburi. I also found freshwater spawn in the lake, in the dam, and near the Burapha University. It will be interesting if you have the student.
Okay.
Thanks.
Thank you very much for your explanations. I think we can look forward for your outcomes in the breeding in the futures from your teams, or even from Burapha universities. And then thank you very much for your sharings and then present of your topic today. Thank you very much. Dr.
Thank you.
May I move to the next topics from Ajahn Sapanand Prathong. He is the head of the Coral Reef and Benthos Research Unit from the Department of the Biology, Faculty of Science, Prince of Songkhla Universities. I think he is one of the well-known researchers in Thailand who working in the area of the marine biology, coral reef, and benthos. And sorry to inconvenience to invite you last time, and then this time we would like to invite you to present again about the topic of the biodiversity of the marine polychaete in Thai waters. If you are ready, please start for your presentations, Ajahn. Can you unmute your microphone?
Okay.
Thank you very much.
ครับพอดีไม่ได้พูดภาษาอังกฤษมาเป็นเวลานานมากไม่แน่ใจ Okay, good morning everyone. I'm going to present to show some idea, actually not actually the results of the whole project, but to show some very useful information in terms of the biodiversity of the benthos, especially the polychaete in Thai waters. Yeah, actually, my research unit is not only coral reef, but you can see in my research unit, it is the coral reef and benthos research unit is established for more than 20 years, and we continue as working for the benthos since I stay in the Prince of Songkhla University, and especially with my wife Jintana Prathong, and she has a company, Marine Ecosystem Management, and this is a good opportunity for her and for us to collect the benthos in many places in Thai water, both in the Gulf of Thailand and in the Andaman coast of Thailand. And we have our staff that working with us continuously for more than 10 years. They are very keen with the benthos. So we have many group of the benthos in our collection, not only polychaete, but we have another crustacean and other benthos. But today I'm going to show especially on the polychaete first. So this is our opportunity for the last let's say for the last 15 years that we concentrate very much on the benthos, especially in the Gulf of Thailand, because we have a lot of opportunity to collect the bentos from the Gulf of Thailand, especially in the economic zone of Thailand. That is, the water depth is about 70 meters, and it is about 150 kilometers offshore from the shoreline. It is a petroleum concession area, especially in the Chevron area, and in some concession area in Songkhla Sea that is belong to the CEC International. So we have that opportunity to correct the bentos continuously for every year for more than 10 years. We have some work from the artificial reef and from Pak Bara Bay and Hat Chao Mai National Park, and especially when we're doing the project to nominate the Andaman Sea Nature Reserve to the World Heritage. So we have an opportunity to collect many bentos from many national parks in the Andaman Sea. So we have lots of specimens in our lab. This is the amount of the bento scrap that we collect during the last 15 years. We have more than 4,500 scrap in 15 years. We have two sizes of scrap, the normal scrap, that is 0.1 square meter scrap, and small scrap. The small scrap are from the Chevron concession area. This is the voluntary work for the long-term monitoring of the Chevron concession area. And the normal scrap, that is in blue color, the normal size of the scrap. So we have lots of opportunity to correct the bentos. That is why we have lots of information in many areas. Like this, the opportunity to correct the bentos in the petroleum concession area, that we normally don't have very much opportunity for other scientists to correct in such area continuous for more than so far it's about 14 years already in the petroleum concession area. In some area we use the scuba diving and use the shovel to get the sediment and put in the soft black, and then we carry on and do the sediment sieving on shore. And we also have many opportunity to collect bentos from the artificial reef area, especially when we starting the evaluation project from Pattani and Narathiwat area. When we have the train bogie to be an artificial reef, and also the artificial reef farm from the Department of Fishery and Department of Marine and Coastal Resources. So this is also another opportunity that we have that we collect the bentos. In some area, like in Songkhla petroleum platform, we have a very good opportunity to correct the bentos, especially the sessile bentos from the jacket. And in some area, we have opportunity to use the satellite, to use the ROV photo, and some opportunity to correct the bentos from the 10 meter to 20 meter depth. So, and because our protocol is, we have a special protocol to correct the bentos, and we carefully sorting the bentos from the sediment. We collect all of the sediment in the field to the lab and sort it under the stereo microscope. Sort the bentos and divide into groups into the coracoid, crustacean, mollusk, echinoderm, and other bento species, and then pass it to specialized researcher in our team, because each of them will concentrate on particular group of the bentos. For example, the coracoid, we have like 3 persons to work for each family of the coracoid in the sediment that we collect. So this very actually we like in this photo, we use the tray and the sediment will be sorting very carefully, and we use the QA QC technique to determine, to collect actually we set up at the 95% 85% efficiency. So it means we can correct every single specimen from the sediment. But in some methodology, they just correct the specimen in the field and throw away the sediment. But now in our lab, we collect every sediment, come back to the lab and sorting in the lab. And we have a very good stereo camera actually in our lab in my house. Actually, this is a Chintana. She works all day and night. We have a good camera in our house. So in the next step, what we found now, we have more than 300,000 specimens belong to more than 2,500 species of benthos in our collection. And we found about 30 to 40% are identified species with a lot of purpose of new species. And I think in the future, we are going to prepare a book of the polychaete of Thailand. And we hope that we can organize a workshop for polychaete identification, not only for Thai, but also for international workshop for the identification of the polychaete on the Gulf of Thailand. So every work we set up a database. Like here we have 2,518 species of benthos in our collection, and probably less than this because some of them might be the variation of the same species. But in our lab, we use we have a coding system. If we cannot identify them, we assign them as a species 1, species 2, species 3, species 4. And if we find the same species, same characteristic, is the same one, so we use the same name. So in the future, and even now, we start to doing some identified species. And we are going to do the database of the benthos in Thai water, both from the Gulf and from the Andaman coast of Thailand. So this is some form of the work. And we found that actually the diversity of benthos in Thai water is very high, especially in one particular area like in Pak Mala Bay. We have studied the benthos from the salt bottom community and also the benthos from the coral reef area. And we found, especially in Pak Mala Bay, you can see we can find like a density of 8,000 polychaete per 1 square meters. And we found totally 426 species of benthos in Pak Mala Bay. And I think this is very high abundance and high diversity of benthos. And it's even more when we study the benthos in the coral reef area. In the coral reef rubble, coral reef sediment. In the coral reef area, we found, I think, more than 600 species from the group of the island near the Buron Island. We found more than 600 species of benthos in the coral reef area. And now we just 3 years ago that we try to identify and describe new species of the benthos from the Songkhla Sea. You can see this is our permanent station. We have 8 stations starting from 3 stations close to the shore. Station 1, 2, and 3 in the 3 kilometers from the shore. Station 4, 5, and 6 is about 10 kilometers offshore, and 7 and 8 is about 30 kilometers. And you can find the impact from the sedimentation and runoff from the Songkhla Lake. And you can see the color of the water, the high sedimentation along the near shore station. And we found the number of species are variable between stations according to the depth and the influence from the Songkhla Lake runoff. You can see that we found in station 7 and 8, it's more than 700 and even nearly 800 species of benthos from the clear water. However, in the disturbed area close to the shore that have a lot of sedimentation, a lot of the sedimented tubation, we found like 400 species, but it's still high number of species compared to other study. And we starting to describe now from this area, we have like a 13 new species of the polychaete from different genera, different family of the polychaete. This is some from this area, from the Songkhla Sea, we can call Songkhla Sea. We found like 1,500 species of the benthos, and we can identify like Annelida. We found 700 species of Annelida of the polychaete, and we can identify 2 species. Only 135 species that can be identified to species level, but we have like 500 species that we can identify into genus level, and most of them we can find many of them are undescribed species, and we are going to do it day by day. Okay? But still, you can see now we concentrate on only Annelida, but we still have other arthropoda that has lots of species of the arthropod, especially the crustacean in the Songkhla that need to be identified. And show you some example of the species that we describe from the Songkhla. This is actually 2 genus. We describe 2 genus in one paper because it is belong to the same family at the family level. So in one, we do not describe one species for one paper, but one paper more than one species, more than 2 genus, but normally in the same family. So this is also another 2 genus, one genus. One of them are name of my dad. One of them are Walmart with a sponsor for this project, assistant. And this is from Songkhla also, and then it's named for 2 of my research assistant, my sister, and also the name of the Thailandica. And this is nearly the recent one. This is also actually we still have some more Synapsidae, but we can only find some complete specimen and enough specimen to do the morphometric measurement to describe the species. But we still have some more of these Peter Synaptis in our lab that will be described later when we have some more specimens. This is a recent one from early of this year. Another 2 new species of the ancestral cells. I think we are going to declare another set of the ancestral cells species to describe very soon. It will come very soon. So and then you can see that we start identify the specimen from the Songkhla Sea first, but we still have many specimens in this area, in the petroleum concession area, but going to be described later, right? So I just want to show what we have so far in our lab, and we still need more time to do some more work on the benthos in Thailand. Actually, the benthos, especially the polychaete, is very large group of the bentos. Because in many crab, we found like 40 up to 40% are polychaete. Another 40% can be crustacean. So we actually we have to say that we have lots of very high diversity of the polychaete in the Gulf of Thailand and also in the Andaman coast of Thailand. Thank you very much.
Okay, thank you very much, Ajahn Sakanan. I think you are the one that work a lot in the bento samplings in Thailand and a lot of the publications that you publish about the new species. That is very much appreciated on these ones. We still have the time to discuss and then ask about the questions. We have the one from the on-site in this conference room. Professor Thamasak, please.
Yes. Thank you, Mr. Chair, and thank you very much, Ajahn Sakanan, for your talk and sharing information about the polychaete biodiversity studies in Thailand. And I have to say congratulations for Pla Thong family for several new species that you collect and describe. And I think that is very important aspect for marine biodiversity study in Thailand. And if you have many new species, I think that is the point that we can show the people that we still need more efforts for the marine biodiversity studies in Thai waters. And what I'm going to share with you is that are there any polychaete taxonomists in Southeast Asian country that we can work in collaboration with them?
I'm not sure. There are some group from Singapore and Vietnam and Indonesia as well. But from our specimen now, because we work directly with the researcher from each group of the genus and family. So we have working with the professor from Mexico, Dr. Rari from museum ที่อเมริกา and from Spain แล้วก็ Dr. Pat from Queensland Museum. I don't know anyone in the Southeast Asia.
Okay, thank you very much. So you should be the best one in Southeast Asia. Anyway, thank you.
No, not me. Actually, my wife. my wife. This is only her work.
Okay. Thank you very much for the questions and the explanations. And then one point just would like to know about the overviews, because you work a lot in terms of the bento sampling in Thailand. And then can you share us about the trends, like abundance trends from your data or information, like in the future trends or on the past, what are the trends of that abundance or the community species in Thailand now?
I think we still have a lot of group of bentos especially in the Gulf of Thailand that need to be described in the future. But we don't have much researcher to concentrate. Most of us are working at the family level, but when we try to work at the genus level, we don't have enough researchers. But if we work directly with the real expert in each genus, so we can show, even we show them in some genus there only one or two species and very different from the specimen from the Gulf of Thailand. So it should be still have many work for the polychaete, not only in Thailand but in the world as well.
Yeah, I think we
a very big group of the research for the polychaete in the world.
Okay, Yes, that is the one things or important points from this conference as well to connect or more collaborate with the partners in ASEAN or the other countries. And then I think from your presentations and information, it's very good to start to the connection with some of the other researchers in the futures. Once again, thank you very much, Ajahn Sakanan Patong, for your presentations, which is very good. Thank you very much.
Thank you. Thank you very much.
Okay, then we will move to the next invited speakers. Will be from the Dr. Ladawan Sangsawang, Department of Marine and Coastal Resources, Thailand. Dr. Ladawan graduated from the Graduate School of Science and Technology Educational Divisions, Department of the Environment and Energy System from Shizuoka University, Japan. And now she works for the seagrass ecosystem projects in the Eastern Gulf of Thailand and surveys, monitoring, restorations, and carbon stock analysis in the seagrass habitats. And today we are pleased to welcome Dr. Ladawan to present about her topics on the current status on seagrass and blue carbon research in Thailand. Dr. Ladawan, please. Sorry, we cannot hear you well. Can you check your connections or the equipments? Dr. Ladawan, we cannot hear you. We still cannot. Okay. Not yet. Maybe. Maybe you can check the system, the sound system, but I'm not sure what happened in your site. And then we can come back in your section again, or no. Okay, we can move to the next speaker first, and then we come back to Dr. Ladawan again. I think we have next topics from the on-site presentations. He is our faculty member as well in Lakamhang Universities from the Dr. Vichin Suparas. And Dr. Vichin Suparas, he received a PhD from the Environmental Science graduate program from Chulalongkorn Universities, and then he is now the lecturer at Lakamhang Universities. And he working in the several international organizations, and also his expertise on the marine ecologies and biodiversities Today he will talk about the 30 by 30 targets, a way to achieve biodiversity goals in Thailand. Dr. Vishin, if you are ready, I mean, you can start your presentation.
Thank you, Mr. Chairman. Can you hear me okay?
Yes, very well. Thank you.
Okay, thank you. Today, I'm going to talk about the new target, actually, that is still under discussion on the global biodiversity framework. This is the target for the marine, the protection area. My presentation is on a way to achieve biodiversity goal in Thailand. Okay, so I'm going to start with the general big picture of the global marine biodiversity and
Sorry
their
to interrupt. Can you share
importance.
your
Sorry?
screen? Can you share your screen?
Sorry.
Yeah. Thank you very much.
Can you see my screen?
Yes.
Okay. So I'm going to start with the big picture of the global marine biodiversity and their importance. And the ocean are large and consist of various types of ecosystems, and they are as a home for various marine species. I search the updated number of marine species that listed in the World Register of Marine Species. I found that until now, there were about 240,000 marine species that they have been described until now. The distribution of marine species also rely on the availability of the ecosystem, particularly coral reefs, seagrass bed, mangrove, and other types of coastal wetland. So based on the distribution data of fish, marine mammal, and seabird, we have at least 6 regions of the marine hotspot. So the hotspots are mostly distributed in the southern hemisphere and include marine areas in temperate and tropical region of Atlantic Ocean, Indian Ocean, and Pacific Oceans. Okay, here is the distribution of the diversity of marine species across the Asia Pacific, and you can see there is lots of species with the large biodiversity in that area. So all level of marine biodiversity promote the provisions of marine ecosystem functions, and the process such as taxon representation, recovery rate, productivity, stability, and resistant disturbance. From there, a good condition of ecosystem function and processes provide a diverse ecosystem service. This service is really important, linked to the human well-being. This emphasizes that the importance of biodiversity conservation to maintain the quality of ecosystem function, processes, services, and securing the provisions of ecosystem services. So, however, the global assessments reveal the risk of extinction in marine biodiversity. Based on the records, about almost 13,000 of marine species were assessed by the IUCN in 2018, of which about 10,000 of them in the category about 10,000 of them are in the category of other than data deficit. The number of the assessed marine species is extremely low, compared to the number of the species described in the worm database. So of the assessed species, about 11% are either critically endangered or vulnerable, and are thus considered threatened with an extinction. So if we look at the marine species threatened with the extinction that may were assessed with the critical endangered and vulnerable. So bony fish, and you can see that they has a higher proportion followed by anthozoans, cartilaginous fish. So change in biodiversity and ecosystem services can be driven by both direct and indirect drivers, consist of land use, introduction or removing of species, adaptation, the use of technology, or even the climate change. For the direct drivers that also influence from indirect drivers, such as demographic, economic, socio political change, scientific and technological change, as well as the cultural characteristics. So what is the marine protected area? So marine protected area is one of the components under the protected area that defined by the IUCN since 2008. They said that a protected area is a clearly defined geographical space recognized, dedicated, and managed through legal or other effective means to achieve the long-term conservation of nature with associated ecosystem services and cultural values. The protected area consists of 7 categories. The first one is a strict nature reserve. The second one is wilderness area. Follow that is the national park, natural monument of features, habitat species management area, the protected landscape or seascape, and the final category is the protected area with the sustainable use of natural resources. So why do we need to have a protected area? So protected area, even the terrestrial and marine, provide a wide range of benefit of ecosystem services. I like to focus on the marine protected area. There are benefits that we can get from having the protected area. The main reason or the main features of the benefit that we can get from marine protected area is that the marine protected area is the conservation area for biodiversity and the ecosystem. This is the thing that can support and maintain the quality of ecosystem services that we are rely on. Besides the conservation of biodiversity and ecosystem, we are also beneficial from many things from marine protected area, just like it's a source of fish population, activities, critical breeding grounds, nursery grounds, and even the feeding habitats. The marine protected area can be used as a marine tourism area that can generate the economic outcome to local community and the nations. It also provides a place for education, training, heritage, or culture, and for the long-term research. And last but not least, for the climate action, marine protected area play a key role in sequestering the carbon dioxide from the atmosphere and storage storage them in several forms. So this is the marine protected area with a wider ocean governance. If we if we look at the governance, we can see the spectrum, right? The spectrum of use and the spectrum of the health of the oceans and the benefits to people. And you can see that the marine protected area provide the area that have a different intensive utilization. Even the no-take area, the multiple use area, and the wider ocean. And you can see the gradient of the healthiest oceans and benefit to people. That has concentrated on the marine protected area. And marine protected area can also regulate the act. Here, I'm going to mention about that the earlier target, which is called IG target, that has been implemented 10 years ago, and now the global community is trying to create the post 2020 global biodiversity framework, which indicate and construct the new indicators or even the target for the conservation, for the biological conservation for the globals. Here, a global biodiversity framework is still under discussions for the period for the period of time starting from last year and until this year. They are going to set the target for the 2030, and you can see that the IG target, if we look at the marine protected area in IG target, they set about 10% of coastal and marine areas to be protected or to be conserved. But for the global biodiversity framework, they increased the target from 10% to 30% to be achieved in 2030. If we dig down into the global biodiversity framework, uh, until now, they are considering the first draft. And if you see that they have a total of 21 action-oriented targets, which cover about 3 themes. The first one is reducing threats to biodiversity. The second one is talking about the sustainable use and benefit sharing. And the third one is the tool and solution for implementation and mainstreaming. If we look at the target 3, which is focused on the increase of the MPA or the terrestrial protected area, they are focused on the achievement of the area of both land and sea area to be protected for 30% in the next 10 years. And this target is still under discussions. They are going to discuss more, and they will be adopted in the CBD COP next time. I think the next CBD COPs will be held in Ming around the end of this year. I'm going to share with you about the outputs and the key point from the discussion from the post-2020 Global Biodiversity Framework working group and the meeting of the subsidiary body on scientific, technical, and technological advice, or SABTA, which is held on at the 24th time. These meetings were organized in Switzerland during 14 to 29 March this year. They have debate on the definition or even the contents of the target 3, right? And so far, many countries tend to support the 30 by 30 target. For example, Canada, the US, the UK, or even the European Union. And in Asia Pacific, you have New Zealand, Australia, Japan, Korea that support the 30 by 30 targets. And in Southeast Asia, we have the Philippines and Vietnam that show their intention to support the 30 by 30 targets. For some countries, they support the 30 by 30 target with the comment about the inclusions of the indigenous people and local community in the MPA, right? And as I mentioned earlier, the global biodiversity framework will be more discussed during the working group meeting in Nairobi next month. And there would be a clear and concrete framework after that. And now I'm going to show you about the global status of protected area. I search from the website from the database from the UNEP WCMC, which is the United Nations Environment Program, World Conservation Monitoring Center. And we found that so far for the terrestrial protected area, we have about 40%, 50.79% of the total land area. If we look at the marine protected area, now we have 7.9%, right? And if we put the another categories of the conserved or protected area, we call it OECM, right? The proportion is a little bit increased. And here is the regional status of protected area, right? And in the Asia Pacific, we have a total with 35,000 locations of protected area. I'm going to bring you all to our countries in Thailand, right? Thailand have a total area of about 23,000 square kilometers, which in the Gulf of Thailand and the Andaman Sea. For the Gulf of Thailand, we have about 200,000 square kilometers, and in the Andaman Sea, we have about 120,000 square kilometers. That include internal waters, territorial sea, contiguous zones, or even the exclusive economic zones. For the establishment of Thailand's marine protected area, we have several authorities that have the law that can designate their own protected area. The Department of National Park and Wildlife and Plant Conservation can designate the marine national park, wildlife sanctuary, no hunting areas using their own law, the National Park Act, the Wildlife Conservation and Protection Acts, right? The Department of Fishery can use their Royal Fishery Ordinance to designate the area for aquatic plants and animal conservation. The Office of Natural Resources and Environmental Policy and Planning, or ONEP, can designate the environmental protected area under the Enhancement and Conservation of Environmental Quality Acts, right? ONEP can also have focal point of Thailand for Ramsar conventions to propose locations to be designated as Ramsar site, as well as the DMCR, which is a national focal point for the UNESCO Man and the Biosphere program. that can designate, that can propose the biosphere reserve. Also, the DMCR has their own law, which is the Marine and Coastal Resources Management Promotion Act, to allow them to designate the area in marine and coastal to be a marine protected area. Here is the current status of the MPA in Thailand. So far, we have a total area of the MPA of about 15,000 square kilometers, which is account for 4.774 of the total Thailand's marine areas. Most of them are the marine national park and the area for aquatic plants and animal conservation. Here, as I mentioned earlier, the Department of Marine and Coastal Resources can establish marine protected area under the Act on the Promotion of Marine and Coastal Management. During the past 5 years, from 2016 to 2020, about 20 areas in 15 provinces have been prepared to be designated as an MPA, covering a total area of 11,000 square kilometers. That means in the future, if we have a total area of this targets, we will be accounting for 8.2% of the total marine area. So, just like I mentioned, here is the state of the number of the MPA that lies at each process to be designated as the MPA. For the next step of the MPA implementation in terms of the DMCR, by the end of the 2032, DMCR aims to protect about 20 locations of key marine and coastal areas, 10 locations for coral reef, including the underwater pinnacles, islands, 5 locations to protect rare endangered marine species, 1 seagrass bed, 2 islands, 1 mudflat, 3 beaches, and 1 artificial reefs. And here is some strategies to increase the MPA in the future. Among the scientific community, we are discuss this, and we suggest and discuss the way or the solution to increase the MPA in Thailand. Some of the possible strategies that can increase the MPA is that we may include the temporary fishery closures, but this one is still on debate. We also include the coastal waters for that reserve for small-scale fisheries. We can use the tool for OECM. We can designate as the locally managed marine area offshore, or we can seek for the marine area in offshore area. Even we can find some collaborations or cooperations with other countries to get the marine protected area in high sea. As I mentioned that in the target 3 of the global biodiversity framework, they mentioned about the OECM, which is the other effective area-based conservation measure, which is one of the tools that can help increase the protected area. For these photos, it shows that if we include all of the strategies that we mentioned, right, we can achieve about 20.1% of the total marine area of our countries. So, what is the Thailand decision on this? This is not. There is no official statement from the Thailand government, but speaking among the scientific communities, policymakers, we tend to agree to adopt the 30 by 30 target after the adoption of the CBD COP15, which is held in Kunming um, around the end of this year, right? But we still have some key challenges in right in this target, because 30 by 30 is a new issue and the new target. People would are concerned about the large proportion, right? The large proportion of the marine protected area that we are going to achieve or not, right? And establishing MPA is time consuming, right? Uh, for each area that we are going to designate, it takes a lot of time, right? We have many processes. If we have more tools, if we have more area that can be designated with a short enough time, that would be a great benefit to Thailand, right? Another key challenge is that when we talk about the MPA, there would be some conflicts among the resource user or even the governments with the resource users. They are concerning about having an MPA might affect the local livelihoods or even the benefit that they are gaining from the area. Um, Last but not least, the key challenge is that some sectors or some agency have a misunderstanding of the target, the 30 by 30 targets, and this would be overcome with having more dialogues or conversation together to make it more understandable together. And I think this is all of my presentation, and I would be happy to answer your question and sharing some idea. Thank you very much.
Okay, thank you very much, Dr. Vishin. I think this is one of the good informations and presentations that we can know about the update targets or the issues that relate into the marine ecosystems, and also the situation in Thailand as well. That is important to learn in your presentations. And then we still have some time to have some discussions with Dr. Vishin. I think we have one from the on-site conference. And Dr. Makamat, please.
Yes, thank you, Chairman, and thank you, Dr. Vishin, for the very nice presentation. Yeah, for my question is a short question. Are there any negative impact if Thailand commit to the 30 by 30 target, but cannot reach the target?
Yeah, thank you for your questions. And this question is the general question that people would ask, right? For me, I would say no, right? If we look at the IG target, the targets that require 10% of coastal and marine area to be protected, most countries did not achieve the target. The global value of the marine protected area is only 7.9%, and Thailand has only 4.7%, which do not achieve the target, although they have put a great effort on this. There were not any negative impact or any punishment if you didn't meet the target. In contrary, I see some benefit from meeting the targets. At least Thailand can show the intention to the world society that we are continuously working. We don't ignore about the MPA expansion to protect biodiversity. Thank you.
Thank you, Dr. Vishin.
Thank you very much. And then one of my point that we know that we have a global goals, SDGs. I'm not sure, are there any relations between this target and SDG goals? And can you explain about this one a little bit?
Yeah, for the post 2020 global biodiversity framework has actually derived from the global sustainable the development goals, right? They bring some of they bring all of the SDGs to be adopted for drafting up the zero draft or the first draft. Anything that see in the first draft is derived from the SDG.
Okay. And the second of my point that is about in Thailand, if we try to implement this kind of the target, who should be the focal stakeholders in these criteria or the targets? Can you share the experience or your opinions on this one?
Right. The I think the authority that should be in charge with this kind of thing is the Department of Marine and Coastal Resources, the Department of National Park, Wildlife and Plant Conservation, that they have their own law to designate the marine protected area. And we may have some others agencies, even the Department of Fisheries, that they can have their own laws. And if we have to if we need the different types of MPA, right? Just like OECM that I mentioned, we may find out some authority to be in charge or to be like a coordinating body for the relevant agencies to do so.
Thank you very much. That is very clear pictures for your topics. And then I think this is quite relevant in the future as well for our Thailand situations. Thank you very much again, Dr. Vishin, for your presentations. And
very welcome.
Yeah. Okay. Maybe we'll move to the presentations of Dr. Latawan again. I think now she is ready to present. And then Dr. Latawan, please.
Hello, everyone. Yeah, first.
Yes, very clear now. Thank you.
Okay. Now I can present now.
You can share screen and you'll start presentation now.
Okay,ค่ะ. Is it show in full screen now?
Yes, now in the full screen. Thank you.
Good morning, everyone. My name is Ladawan Sangswang. I am a marine biologist at Department of Marine and Coastal Resources. First, thank you, Dr. Thamasak Yimin, to invite me to here. Today, I'm here to talk to you about the current status on seagrass and blue carbon research in Thailand. Seagrass meadows are among the most productive and important ecosystems in the world. They are found in shallow salty and brackish waters. Seagrass are marine flowering plants that support thousands of key species and essential ecosystem functions, such as improve water quality, protect coastal line, cycle nutrients, and create habitat corridors between coral reef and mangroves. Sometimes seagrass referred to as the lung of the sea because they are photosynthesized to create energy and grow and absorbing carbon from the water and generate oxygen in the process. In addition to absorbing carbon in their leaf and root, seagrass also trap decaying organic matter and silt, creating carbon-rich sediment, making them very effective carbon sinks. Now, seagrass threats are more complicated than you think, especially in the developing countries. In Thailand, we lose seagrass area from human anthropogenic activity, such as developing, coastal developing, tourism, dredging, aquaculture, and bloat effects. And we also lose the seagrass area from the natural, such as from season change, large ocean grazing events, and phytoplankton blooms, caused to reduce the light to the bottom. Thailand has 257 square kilometers of seagrass base. There are 86 square kilometers in the Gulf of Thailand and 170 square kilometers in the Andaman Seas. The composition of seagrass in Thailand now has 13 seagrass species and seagrass coverage. Percent of seagrass coverage about 40 to 60%. The dominant species is Enharat acoloides and Halophila ovalis. This slide shows the distribution of seagrass along the coast of Thailand. And in the eastern Gulf of Thailand, the large seagrass area is อ่าวมะขามป้อม อ่าวคุ้งกระเบน แหลมกลัดไม้ลูด and for the middle Gulf of Thailand is อ่าวทุ่งคาสวี อ่าวบ้านดอน เกาะสมุย เกาะพะงัน and for the Andaman Sea the seagrass area is อ่าวบางเบน อ่าวทุ่งนางดำ เกาะพระทอง อ่าวปากข้อ เกาะสีบอยา and เกาะลิบง which Got Libong is the largest seagrass area in Thailand. They are located in Trang province, and the black symbol is show the 7 marine institutes belonging to the MCR. They are along the coast of Thailand. The responsibility to monitor, surveys, and research about the marine resources, such as coral reef, marine mammals, seagrass, and marine biodiversity in different parts of Thailand, such as the Eastern Gulf of Thailand, the Upper Gulf of Thailand, the Middle Gulf of Thailand, and the Andaman Sea. And we collect the data from our institute for making the country baseline data and marine resources annual report. This slide shows the 13 seagrass species in Thailand. We separate in 3 sizes. The big size is Enharat acoloides, and the medium size is Cymodosia cerulata and Cymodosia rotundata, and the small size is the genus of Halodu and Halophila. This is according to the survey in 2021. The total of seagrass in 2021 is 99,326 light, or 158 square kilometers, and or 62% of the potential seagrass area in Thailand. We found 12 species except Lupia maritima. The status of seagrass observed in 2021 was mainly in moderate condition, 52%, and dense to very dense condition, 28%, and light condition, 20%. For the seagrass change compared with last year, the mainly is stable to enhance 65%. And the degraded by natural is 32% and degraded by the human is 2.5%. The increase of greenhouse gas is known to cause climate change, especially carbon dioxide from anthropogenic activity. Understanding the carbon cycle and how much carbon is stored in various ecosystems such as seagrass bed is very important to help to slow down climate change. This slide shows the global carbon cycle such as carbon dioxide released into the atmosphere by respiration, by decomposition in soil and deep sea, by human anthropogenic, as deforestation, burning fossil fuels. The forest is the world's largest carbon collector. Generally, there are more than 2,300 billion tons which accumulate in its plants and in the soil. Plants can absorb carbon dioxide from the atmosphere by the photosynthesis process of leaves to create organic carbon matter which contain carbon as a component and accumulate in different parts of above ground and below ground. Which plant can release carbon dioxide into the atmosphere to the respiration process. This study points out that coastal ecosystems such as seagrass, salt marsh, mangrove have the potential for carbon storage than tropical forests in a reserve now, or referred to blue carbon. In this slide, I will show some research about blue carbon in seagrass of Thailand. 2012, Anjana Prateep studied seagrass beds as a carbon sink in the Biosphere Reserve and Hat Chao Mai Marine National Park. The results show an important role of seagrass. The results show that the total amount of carbon stored in seagrass at Hat Chao Mai more than at Ranong Biosphere Reserves. 2016, สีน้ำเงิน ปัทมาสีน้ำเงิน study seagrass carbon accumulate at Phungkaben, Chanthaburi province. The results show that the organic carbon in Enhalas Acorides was different between above ground and below ground, while Halodu pinifolia was not different in all parts. The amount of the organic carbon in this type of seagrass was not based on the area of their growth. 2017, carbon content in the Malika Sankovic studied carbon content in different seagrass species in Andaman coast of Thailand. The results show the big size species, Enhalas acorides, have higher carbon content than smaller species, Thalassia hemtrichii and Halophila ovalis. 2018, Pathama สีน้ำเงิน studied the role of blue carbon in seagrass at the east coast of Thailand. The results show that, first, the highest ability to absorb carbon found in Enhalas acorides. Second, the highest organic carbon in soil was found in Phungkaben Bay and Kok Ladat. 2020, Chan Singkhorn studied organic carbon content in seagrass and sediment in seagrass bed at Chonburi province, Sattahip. The results show that the organic carbon in seagrass and sediment was different by season and sediment depth. The organic carbon in seagrass sediment was higher than non-seagrass area. And 2021, Sankovic studied quantification of blue carbon in seagrass ecosystem of Southeast Asia. and the potential for climate change mitigation. The results show that the average value of the total carbon storage within seagrass meadows of this region is 121 megahectare, and the total carbon stock, the total organic carbon stock of the seagrass meadows of these regions was 429 telegram. The result of the study highlight and promote the potential of the Southeast Asian seagrass meadow to national and international agencies as a practical stream for nature-based for the climate change mitigation. And next, for the DMCR, I will show the blue carbon research by DMCR. This picture shows the field work of the collect data from Got Libong. And for the methods of the organic carbon analysis, first, after we collect the sediment by pore, we will measure the length of the soil that we collect, and then we cut in every 5 centimeters. After that, the sampling was weighed and kept in the oven at 60 degrees Celsius, around 72 hours. And then the sampling was divided into 5 grams and burned at 450 degrees Celsius at around 9 hours. And after that, we will weigh the sample again and record the data and bring it to calculate for the organic carbon. And this is the field work for the collecting seagrass sample. And for the method is we will collect seagrass by the contact, and we separate the size of seagrass. For the small seagrass that I mentioned, Halodu, and for the Halophila, we use the small contact, 25 centimeters squares. And for the big size, like Enhalas, we use 50 centimeter squares. When the sampling was cleaned by removing sediment, shell, and the other is make that our data is over carbon. And then the sampling also do the same with sediment. We was weighed and kept it in oven at 60 degrees Celsius, 72 hours. And the sampling was divided into 5 grams and burned at 450 degrees Celsius at 8 hours. For the methodology, we follow from the books Blue Carbon in Seagrass Ecosystem, Guideline for the Assessment of Carbon Stock and Sequestration in Southeast Asia. And the method is for that method we call the loss of ignition. This is now Marine and Coastal Resources Research and Development Institute by the MCR. We also prepare a guideline for the assessment of carbon stock and sequestration in seagrass of Thailand, and now it's in process. And this slide shows the results of carbon content in seagrass. The green bar is the above ground and the red bar is the below ground. from this graph show the seagrass have a different ability to store carbon according to each seagrass species. The highest carbon content was found in Cymodocea rotundata and Cymodocea cellulata. This slide shows the potential for carbon sequestration in different areas with different species, composition, and seagrass coverage. And we will found at the Trang province and Krabi province have more the carbon sequestration than the other area because that they have the large seagrass area. This is the conclusion of the blue carbon data from 2020. Seagrass have 12 species and area about 778 light. The condition almost is moderate. The total biomass from 2000 is 731 ton. Highest biomass was found in Enhalus acorides, Cymodocea cellulata, Cymodocea rotundata, and Thalassiea hempicii, respectively. And for the carbon sequence in seagrass, this is only in seagrass, not included in the sediment, is 2,199, 380 ton carbon. And the highest carbon sequestration was found in Enhalus acorides, 867, 600 ton gram. That's all my presentations. Thank you for your attention.
Thank you very much.
Anyone have questions?
Thank you very much, Dr. Ladawan, for your presence about the current status on the seagrass and then ongoing activity of DMRCs, which is very interesting. And then thank you very much. And then I think we have one questions from the audience in the chat box. And then she asked from the Ajahn Anchani Bunpakop. She asks about if we are interested to study seagrasses, leaf anatomies. Do you know who have the knowledge about that?
Seagrass anatomy. I'm not sure, but if I know Ajahn Anchana Prateep from Prince Songkha University, maybe she know very well about seagrass.
Okay, I think that can be the guide for the further discussions with the expert on that one. Thank you very much, Dr. Ladawan, and also Ajahn Anchani for your questions. We have another questions from the on-site conference from Professor Thamasak.
Yes, thank you, Mr. Chair, and thank you very much, Dr. Ladawan, for your contribution to this Asian biodiversity session. I would like to ask you one question, is that based on the standard method for blue carbon analysis, do we need any equipment for analyzing your samples? I mean some sophisticated machine or equipment that we need for analyzing the blue carbon.
Okay, thank you, Dr. Thamasak, for your question. Okay, can you hear?
Yes, please.
Okay, actually, for the organic carbon analyze, we have three. Now I know we have three methods from the analyze. First, we use the LOI, loss of ignition, by use the fullness. And second, we use the CHN analysis. And third, we use the method of the titration of sample. For the more accuracy of the data, data, normally we should use the CHN analysis. But this machine is very expensive for and is difficult to maintenance.
Okay.
And now for the easy, I think for the basic easy everyone can do, I think is the method of LOI method. But I'm not sure about the how different the accuracy of data. Maybe I will do like a compare from the same sampling and do this is different the organic carbon content with different method.
Okay.
Okay, thank you. Thank you very much, Mr. Chen.
Thank you very much, Professor Thamasak, for your questions, and also Dr. Radawan for your explanations. Okay. I think that all the questions from both in the conference online and offline. And then thank you very much again, Dr. Radawan, for your presentations. Thank you. I will move to the last presentation for the morning sections. I think that is from Dr. Nachapon Sangmani. She is one of our faculty members in Rangkapeng University. She graduated from the Department of the Environment and Energy Systems in Shizuoka University, Japan, as well. And also, she will provide us with the video presentations. And then if you have any questions, maybe you can drop into the chat box at the end, and then we can convey your message or your questions into the Dr. Nachapon again. Okay.
Hello everyone. Thank you for coming to my presentation. My name is Nachapon Sangmani. Now I would like to present my research with the title The Relationship of Myofauna Community and Coral Cover at Mukok Surin National Park, the Andaman Sea. Let's start with the introduction of this research. A subbottom such as sand and rubble is an important component of coral reef ecosystem. Many marine organisms live in a sedimentary substrate. Start from smallest microorganisms, for example bacteria and fungi. Myofauna size from 40 to 500 micrometers, and the largest, bigger than 500 micrometers, is macrofauna. Community of myofauna in coral reef are important for marine ecosystem. They contribute to marine food chain, particularly being as a food source for all juvenile of marine economic species. In this research, I would like to examine the composition and density of myofauna in the coral reef at Mukok Surin National Park. To access the relationships of myofauna density and ratio of live coral per dead coral cover. We have 6 study sites in Mukok Surin National Park with depth range from 1.5 to 3.5 meters. Each study site, I collected triplicate of sediments using PVC core, which has a diameter of 3.5 centimeters. The samples were then transported to the laboratory for identification process. We identify myofauna to taxon level, counted them under stereo microscope. The results show the total myofauna density at the study size. Here, อ่าวไม้งาม show the higher density of myofauna, which is about 77 individuals per 10 square centimeter, and it is significant different than the density of อ่าวผักกาด อ่าวจาก and อ่าวสุเทพ However, อ่าวสุเทพ had a lower density of myofauna, which is about 40 individuals per 10 square centimeter. From the previous graph, อ่าวแม่งาม อ่าวช่องขาด and อ่าวแม่ยาย that show a high abundance of myofauna are located in shelter area. Here we found 17 groups of myofauna. Foraminifera, Nematoda, Polychaeta, and Copepoda were a major group for all study sites, and there are some common myofauna such as Tuberculia, Nematine, Ostracoda, Amphipoda, Coleophora, Oligochaeta, Tardigrada, Isopoda, Gastropoda, Scaphopoda, Bivalvia, Echinoidea, and Cypancula. This slide shows the percentage of live and dead coral cover at the study site. We can see that live coral cover are higher than dead coral cover at all study sites. However, the significant difference between live coral and dead coral cover ratio found at อ่าวแม่ยาย and อ่าวช่องขาด Here we also found a positive correlation between total myofauna density and ratio of live coral per dead coral cover. Among 7 groups of myofauna, only Copepoda, Nematoda, and Tuberculia show a positive significant correlation to the ratio of live coral and dead coral cover. Based on the findings in this research, 17 fauna taxa were found at หมู่เกาะสุรินทร์ National Park, and average myofauna density varied among study sites. The spatial variation of myofauna abundance and community structure can be influenced by hydrodynamic conditions. For example, the habitat types in the reef slope of the Maldivian archipelago in Indian Ocean. Myofauna can be also used as a bioindicator for detecting environmental change. Some studies reveal that major myofauna groups such as polychaete, Harpacticoid copepod, and nematode on the coral reef in Brazil show divergent response to the various predict climate change scenarios in reduction of seawater, pH, and increased temperature. In the shallow reef lagoon ecosystem on Great Barrier Reef, Australia show a novel perspective in coral reef trophodynamics between surface sediment myofauna and deposit feeding sea cucumber, and community shift in the future ocean with implication for the functional of coral reef from the bottom up. Our results suggest that the myofauna communities can play a major role in a coral reef ecosystem, and they may be gradually changing in total population density and their composition that may affect the coral reef food web. Finally, I would like to thank SCT 47 Conference, Thai Government Award to Ramkhamhaeng University for Budget for Research Promotion, Marine Biodiversity Research Group, Ramkhamhaeng University, Phuket Marine National Park Operation Center 2, Department of National Park, Wildlife and Plant Conservation, and the last Marine Science Association of Thailand. Thank you for your attention.
Okay, thank you very much. I think we run into the last speakers of today's, and then we have the very good sections for this section in the mornings. And then thank you very much for all invited speakers and participants in these sections. And before we close the section, I just would like to remind you in the evaluation sheet that we drop in the chat box. Our staff drop into the chat box, and then you can check on that one. On this one, we would like to know your feedback so that we can improve or further manage for the conference in the future. And then thank you very much for your response in the evaluation form as well. And before we close the section today, and then I would like to also another request again for have group photos from the online participants. If you are available, maybe can you turn on your cameras, and then we can have the group photos again for the few seconds, please. Okay, maybe okay. 1, 2, 3. Okay. Because I cannot smile. Okay, I cannot encourage you to smile as well. Sorry for that. Okay. Thank you very much for all again. And then the last announcements for today's, and then we will have another sections. We will start in this afternoon, 1 p.m. The panel discussions on the sustainable fishery management in marine protected areas. And then we will discuss in the panels again in the afternoon sections. And thank you very much for your all participation in these morning sections. See you again. Thank you.
Recording stopped.